My name is Emilie Hultin, and in this lecture I will talk about next-generation sequencing in HPV research. I will describe some principles and practices of metagenomic sequencing in HPV research. I will give one example for a specific study on condylomas, and then talk about some ongoing research on the HPV transcriptome. I will also talk about the procedure that is used for HPV testing of HPV-negative cancers in the clinic. So this study was published in Virology, and it's about condylomas. Does human papillomavirus negative condyloma exist? So condylomas is anogenital warts, and this is a sexually transmitted infection with human papillomaviruses. And there are more than 200 different types of HPVs, but the ones causing condylomas are some of the low-risk HPV types, and the most common ones are HPV 6 and HPV 11. Usually, condyloma is diagnosed visually, and it may occasionally be confused with molluscum, which you see here on the bottom of the slide, compared to the upper part here where you see condyloma warts. So in Sweden, we have a condyloma reporting system. And in the southern part of Sweden, there were 621 subjects with condylomas that were reported between the year 2006 and 2009. And these 621 subjects were typed for HPV, and the study was published 2013 by Sturegård et al. Out of these 621 subjects, 94% were HPV positive. However, there were a few that were seemingly HPV negative, and these were also tested with a broad HPV primer PCR, and this primer HPV PCR was published by Söderlund Strand in 2009, but they were still then negative for HPV. So these HPV negative condylomas, how could that be? There are like 4 different possibilities. Could it be true negativity, meaning that there are some condyloma warts that are not caused by HPV? Or is the sample inadequate? Another explanation can be that the detection methods used are not sensitive enough, or that it exists some novel HPV types that are not detectable by the method used. So we wanted to test these HPV negative condylomas by metagenomic sequencing. In metagenomic sequencing, all the DNA in the sample will be sequenced, and we don't need any prior knowledge of the DNA sequence, meaning that we don't need to design any primers that target specific sequences. So in total, we had 40 HPV negative condyloma specimens. 21 were from women and 19 were from men. These were swab samples from the wart. And the DNA were extracted by a robotic system using beads for purification. All the samples were also tested for human DNA by a PCR for human hemoglobin beta, and they were all positive for human DNA. In order to be able to sequence the DNA in the samples, the DNA need to be prepared. And we used the Nextera DNA library preparation from Illumina. We started with 50 nanograms of DNA per sample, and the DNA needs to be, in the first step, fragmented. And that is done using transposomes, enzymes that cut the DNA and at the same time add adapter sequences to the ends of the fragments. This step is called tagmentation. In the following step, all the fragments are PCR amplified in order to add sequencing adapters to the ends. So in the end, we have approximately 300 base pair long fragments with adapter sequences, including index sequences, unique for each specific individual sample. These libraries were then normalized and pooled together and sequenced on the Illumina NextSeq 500 system. And we sequenced the fragments in both directions, 150 base pairs in forward direction and 150 base pairs in the reverse direction. And this generated approximately 2.7 billion bases DNA per sample. These sequences that were generated, so-called reads, needs to be analyzed. And first of all, they were checked by quality, and all the reads with bad quality, with a Phred score below Q30, were removed. The rest of the reads with high quality were assigned to each individual sample by the index sequence that they contained. And then all the reads were screened against known genomes that are in GenBank, and all the human reads were removed from further analysis. The next step is to normalize all the reads and assemble the reads into contigs. And contig assembly is like a puzzle where on top here, in A part of the picture, we see just a normal sentence. The top sentence in orange, we can read like contigs are contiguous sequences of DNA. But from the sequencing, we just get parts of the sentence. So we have parts of the whole sentence, but they overlap with each other. So it's possible to put them together and made a complete whole sentence. And it's exactly the same thing when it comes to DNA sequences from the sequencing. We have a lot of short sequences that overlap with each other. So it's actually possible to put them together into a long contiguous sequence, a so-called contig. And that is what is done during the bioinformatics. So contigs are assembled, and then the reads are remapped to the contigs. And the reads can then be classified whether they belong to different bacterial genomes or viral genomes. And in this study, we were mostly interested in the human papillomavirus reads. So all the HPV reads were also classified based on similarity to the closest related HPV type in GenBank. In total, the sequencing generated over 700 million reads, and 0.05% were related to viruses. 91% of all the viral reads were HPV reads, 300,000 HPV reads. All those HPV reads were classified, and we detected from those 40 Condyloma samples 75 different HPV types. They belong to both alpha, beta, and gamma genera. And you can see the distribution between how many we found that were established. The green one, 24, 5 known putative HPVs, but most of the types we found, 46, were novel putative HPV types. These belong to beta and gamma genera. And 6 of these novel putative types, we got the complete sequence of the whole genome of the HPV, meaning that we got all the 8,000 base pairs. And we could clone 3 of them, and they were assigned official HPV numbers by the International Reference Center. And the numbers were type 200, 201, and 202. So in this picture, we see a Bayesian phylogenetic tree of all the established HPV types and how they are distributed in the different genera, which is shown here in different colors. And the 3 novel types that were detected are shown here in black in this tree. And you see that they all cluster together with the gamma types. So the results of this study were that 92% of these HPV negative condyloma specimens actually contained HPV from after sequencing. And there were no HPV reads in the water controls. And for a specimen to be HPV positive, we have a cutoff at least 5 reads of one specific HPV type. So the table to the left here show number of positive samples for different HPVs. So 37 of the 40 HPV negative condylomas were positive for HPV. 35 samples were positive for established types, and 30 different samples were actually positive for novel putative HPV types. In addition, molluscum contagiosum, another virus that causes warts, were also detected in more than 50% of the samples. And it is known that it's quite common with co-infection between HPV and molluscum. In the table to the right here, we see all the different HPV types that were detected. And if we just look at the top of the table, we see the most common ones in this study. So the most common one was HPV 6, followed by HPV 180. These two types were detected in 30 samples and 18 samples, respectively. And the 3 novel types that we found and cloned, type number 202, 200, and 201, were also present in several samples. So to answer the question if HPV negative condyloma exists, it's not really clear yet, but however, 37 of those 40 apparently HPV negative condylomas were positive for HPV by metagenomic sequencing. And one of the most common types in this set of samples were HPV 6, as you saw in the previous slide, which should have been detected by those normal PCR-based methods. However, an explanation could be that these samples contain variants of known HPV types, variants that have mutations within primary regions. Another explanation could be also that these samples also contain a lot of novel HPV types that are not detectable by the other PCR-based methods that were used. But still, we have 3 of the 40 samples that are still negative for HPV. And whether they are true negatives or just a misdiagnosis or an inadequate sampling is very hard to tell. It might also be that we need even higher sensitivity in our detection methods. But if you remember, more than half of the samples were also positive for molluscum contagiosum. So any of those samples could be a misdiagnosis there. In the study, 46 novel putative HPV types were detected and 3 are now cloned and established. So then I would like to talk a little bit about HPV transcription in cancer. As all established oncogenic infectious agents persist transcriptionally active in their host, it would be very interesting to look for transcriptionally active HPV in certain cancers. And how can we do that? We wanted to look for RNA from HPV by RNA sequencing of tumors. So we have 3 different kinds of cancer samples. At top here, we have a positive control. This is a cervical cancer cell line called CASCI, and this cell line is known to contain 600 copies of integrated HPV 16 genome, integrated in the human genome. So that could serve as a positive control. Then we have HPV negative cervical cancers. And these are formalin-fixed paraffin-embedded blocks of cervical cancer biopsies. And in total, we used 6 cervical cancer biopsies. And then we have also some skin samples. We sequenced 2 squamous cell carcinomas from fresh frozen skin biopsies. And we did RNA sequencing using the true 6-stranded total RNA library preparation kit, and we sequenced 150 base pairs paired end sequencing. And here are the results from the Kaski cell line known to contain HPV-16. The table to the right shows the number of reads for the different species, like human, bacteria, viruses, etc. And we see in the viruses in green shows that we detected around 18,000 viral reads. And the HPV-16 genes that we detected were the early onset genes E1, 2, 4, 6, and 7. And this is also visualized to the left here, where we have a coverage plot over the whole HPV-16 genome from this RNA sequencing. So in the middle, we see the circle which represents the HPV-16 genome, where all the genes are located. And then the orange bars around it represents number of reads in each position in the genome. And you can see that the orange bars are really high to the right and quite low to the left. So we see that we have a very high coverage on the early onset genes, the E6, 7, 1, 2, 4, and 5, whereas the late onset genes L1 and L2 shows very low coverage. And that corresponds also to the genes that we detected by the sequencing. And the Kaski cell line contains integrated HPV-16 genome, and parts of the genome has then been lost. So it looks like the early onset genes are integrated in the human genome. So then we have results from HPV negative cervical cancers, and these were FFPE blocks. These cervical cancers were HPV typed by a Luminex method that targets the L1 gene of 37 different high-risk and low-risk HPV types. And the table here shows the 6 different biopsies or cervical cancers that we used. All of them were positive for human hemoglobin beta by real-time PCR. Luminex we choose to include 2 samples that were positive by Luminex. Sample number 5 were positive for HPV-18 and number 6 positive for HPV-16. But the 4 samples, 1, 2, 3, and 4, they were negative for HPV. And these were reviewed by a pathologist, and they were confirmed to be adenocarcinomas and invasive cervical cancer. We also performed real-time PCR for HPV-16 and HPV-18 of these HPV-negative cervical cancers. And now sample number 4 were positive for HPV-16. However, the 3 other samples were still HPV negative. Then we sequenced those samples. First, we did DNA sequencing. So we sequenced all the DNA in the samples. And we could detect HPV-18 and HPV-16 in sample number 5 and 6, which corresponds to the results of the Luminex. We also detected HPV-16 in sample number 4, which corresponds to the results from the real-time PCR. In addition, we also detected HPV type 128 in sample number 1 and 2, but sample number 3 were still negative. Then we did RNA sequencing. So we sequenced all the RNA present in the samples. And All samples except sample number 5 were positive for one or more HPV types when we looked at the RNA. And when we looked also which genes that we detected by the RNA sequencing, and it was the early onset genes 1, 2, 4, 5, as well as the late onset gene L2. And interestingly here is to see that we did not detect E6 and E7, which is the target for the primers in the real-time PCR. And we did not detect L1 gene, which is the target with the Luminex typing assay. So that could be an explanation why these samples appeared HPV negative by the PCR-based systems. And now they were positive by sequencing. The last part of this study was that we also sequenced RNA from 2 different squamous cell carcinomas of the skin. So we see the results here from the 2 different samples. And looking at the viral reads, we detected 106 reads that mapped to HPV 110 in sample number 1, and 10 reads in sample number 2 of HPV 110. So in this study, we could detect actively transcribed HPV in squamous cell carcinomas. So as concluding this lecture and talk a little bit about some future perspectives, we can say that metagenomic sequencing is a powerful tool to detect and discover new HPV types. And RNA metagenomic sequencing can be used to detect transcriptionally active HPV infection. We tried this and we detected active HPV infection in non-melanoma skin cancers and cervical cancers, as well as a cell line known to contain HPV. We also were able to do this both in FFPE biopsies of cervical cancer and fresh frozen biopsies of the skin. And the skin non-melanoma skin cancers contained actively transcribed HPV 110. And for the future, this RNA sequencing in cancers can be used for viral transcription analysis, as well as looking for viral integration into the human genome. And it could also be a tool to discover and detect other RNA viruses than HPV.